In this topic, we're going to discuss translocation. So we're going to look at what is translocation, what is a source and what is a sink, and what is the source and sink in a potato plant during the different seasons. What is the structure of phloem, and what does phloem look like under the microscope? So what is translocation? Well, in the last topic, we discussed the transport of water. In this topic, we're going to discuss the transport of soluble products in the plant. So this is translocation. Plants make carbohydrates in the leaves by photosynthesis. They also use some of these carbohydrates to make amino acids, proteins, oils, and other organic substances. The soluble products that a plant uses are sucrose, amino acids, and fatty acids. These are carried to all the parts of the plant in phloem tubes by a process called translocation. Now, the sap inside the phloem tubes contains a lot of sugar, particularly sucrose. So what is the definition of translocation? This is the movement of sucrose and amino acids in phloem from regions of production to regions of storage or to regions of utilization and respiration or growth. So the regions of production would be like your leaves, and the regions of storage would be like in the roots or tubers. Plants also need these products at the shoot tip for growth. So what is the source and sink? Well, the part of the plant from which sucrose and amino acids are being translocated is called the source. And the sink is the part of the plant to which sucrose and amino acids are being translocated. So source is from, sink is to. Here you've got a potato plant that is photosynthesizing and growing. This would happen in summer. What do you think will be the source of the sucrose? Well, the leaves will be the source because they produce sucrose. The sucrose then gets carried in the phloem to all the parts of the plant. So these parts would be the sinks, which include the roots, flowers, and fruit. Now the roots may change some of the sucrose to starch and store it. The flowers use the sucrose to make fructose, which is found in nectar. Later, when the fruits are developing, the sucrose may be used to make those nice juicy fruits. Many plants have a time of year when they become dormant. During this stage, they wait out harsh conditions in a state of reduced activity. So in a hot climate, this may be done during the hottest, driest season. In temperate countries, it may be during the winter. Dormant plants do not photosynthesize, but they survive on their stored starch, oils, and other materials. When the season changes, they begin to grow again. So now the stored materials are converted to sucrose and transported to the growing regions. So here you can see a potato. Notice how it's in a state of dormancy. So let's have a look at the example of potato tubers. During the summer, the leaves of the potato plant can photosynthesize. So the source is the leaves, and the sink is the potato tuber. The sucrose is transported from the source to the sink. So at the sink, those are your tubers. So this is where the cells in the root tubers change the sucrose into starch and store it. So in winter, the leaves will die and nothing's left of the potato plant above the soil. In spring, they will begin to grow new shoots and leaves. So here you can see this would be happening during spring. The potato plant's growing new shoots, and the potato carbohydrates are stored as starch. So the tuber is the source. Starch is going to be converted to sucrose, and it's going to move through the phloem to the growing shoot by translocation. So the shoot is the sink. So you can see from this example that phloem can transport sucrose in either direction, up or down the plant. This doesn't happen in xylem vessels. So xylem vessels only transport water upwards because transpiration always happens at the leaf surface. Okay, let's have a look at the structure of phloem. So here you can see phloem tubes, which are called sieve tubes, and cells sitting next to them, which are companion cells. So together, companion cells and sieve tubes make up phloem tissue. So phloem is made up of sieve tubes and companion cells. Now the sieve tubes are almost hollow, elongated cells placed end to end. They are separated by sieve plates, which are perforated cell walls through which cytoplasm and cell sap can flow. So phloem tubes have got no nuclei and very few organelles. Cytoplasm and cell sap can move from one phloem cell to the other through the sieve plates. Now sitting next to the sieve tube, you've got the companion cell. These are active cells that provide energy for translocation. So translocation is an active process requiring energy from respiration. Okay, let's look at the structure of phloem. So here you can see phloem tubes and companion cells. There is also phloem parenchyma, which are those little cells. They are known as packing cells. So parenchyma are the packing cells. You'll learn more about that at A level. On the left here, you can see a cross-section through the stem. So it shows you the vascular bundle quite clearly. What do you think the part labeled X is? Well, it's got lignified walls, so it must be the xylem vessel. The phloem sieve tubes have smaller companion cells sitting next to them. And if you look at the longitudinal section on the right, you can identify the sieve tube elements quite easily by their sieve tube plates. Here's another image to show you phloem tubes in longitudinal sections. So this shows long phloem sieve tubes with the sieve plates in between. So they have a small amount of cytoplasm around the edges. And then there's a cross section to show you a sieve plate. So it's quite difficult to get a sieve plate in cross section. Okay, finally, companion cells. Unlike sieve tubes, the companion cells have a dense cytoplasm, showing that the cells are active. There's a small vacuole, and translocation is an active process that requires energy from respiration. So the companion cells provide this energy. Here's another diagram to show you phloem under a microscope. So notice the large sieve tubes and the smaller companion cells sitting next to them. And then you can see in the middle there, those lignified walls that have stained red are your xylem vessels. Now, you need to use this diagram to draw and label the diagrams in your notes. So notice the sieve plate, the phloem sieve tubes, and how the phloem sieve tubes do have some cytoplasm around the edges, but they do not have any nuclei or other organelles. And the companion cells have nuclei and lots of organelles. In summary, we looked at what translocation is. So can you define translocation? What a source and a sink is, and what the source and sink is in a potato plant during the different seasons. So remember, it's different during summer compared to winter and spring. We looked at the structure of phloem. So remember that phloem tissue comprises sieve tubes and companion cells sitting next to them. Which one contains organelles? the companion cells, and then we also looked at what phloem looks like under the microscope. And that concludes our lesson. The end.